Okay, so this video we're going to look at neurons and synapses for A2. So obviously this is linked to coordination along with the other bits that we've looked at as well. Obviously we're looking at the nervous system. So the nervous system is made up of neurons or nerve cells which carry electrical impulses throughout the body, and they cross at synapses. Okay, so we have receptors and effectors. Receptors obviously pick up stimuli. Effectors react to that stimuli and cause either movement or glands to release hormones. And you have a coordinator, which tends to be the brain, found in the central nervous system. Okay, the first part of this is looking at the three different types of neurons. So we have sensory neurons, we have connector neurons, and we have motor neurons. These are normal type cells, so they have a nucleus, they have a cytoplasm, they have a cell membrane. Obviously, they are specialized in that they have a long, what's called the axon, which may stretch from down the base of your back to the end of your toes, or up the top part of your back to the end of your fingers. Okay. They also have these swan cells that we'll take a look at that wrap around. They provide an insulating layer around neurons, which allow impulses to travel faster. They have dendrites, which you can see at the end, which allows them to make many connections. And they have synaptic bulbs at the other end of the neuron, again, to make connections with other neurons. Okay, so the sensory neuron is what is linked to your five senses, if you like. So it picks up the stimulus. So this end, just before the dendron, will have receptors for things like touch in the skin, for taste in your tongue, for sound waves, etc., in your ears. Okay, it will carry the message along the sensory neuron. You can see the cell body is just off the center of the axon. Okay, and then it carries that impulse on, and that travels into the central nervous system. In the central nervous system, these synaptic bulbs will synapse with the dendrites of the connector neuron. This is found, as I say, in the central nervous system, so either in the brain or the spinal cord. And these synaptic bulbs will synapse then with the motor neuron, the dendrites of the motor neuron, and carry the impulse to either a muscle, which will cause movement, or a gland, which will cause the release of a hormone. Okay, so if we take a look in detail at the structure of the axon with the swan cells, here you can see at part A, if you take a cross section through there, you've got the axon in the center, it's got the swan cell wrapped around it, and it's got part of the nucleus of the swan cell that you can see there. Part B, again, you've got the swan cell in the nucleus, which you can see here. And then in part C, which is the gap in between the swan cells, which are called the nodes of Ranvier, you can see you just have the axon, which you can see here. Okay, the swan cells are sort of flat cells wrapped around the axon, and they do contain a nucleus, which you can see. There's the nodes of Ranvier in between the swan cells. That's the cross section. Okay, and in terms of how that all gets put together, you can see here we've got a hot plate. Okay, and when you touch the hot plate, that's picked up by receptors in your skin. They will send a message up the sensory neuron or along the sensory neuron, which travels into your, in this case, the spinal cord or your central nervous system. You can see there's a little synapse there between the sensory neuron and the connector neuron, or relay neuron as it's sometimes known, which is found in the central nervous system. And then that synapse is with a motor neuron, which you can see here. In this case, is connected to a muscle, or it can be connected to a gland, and that will cause that muscle to react and contract and move your hand away. Okay. Now, in terms of the next bit, we're looking really at how an impulse is sent. Okay, so the first part of this is looking at the resting potential. So this is, if you imagine your axon, okay, and within the axon, within the membrane, which is a normal bilayer, within the membrane of the axon, you have a sodium-potassium pump, which works by active transport. you have sodium channels, and you also have potassium channels along different parts or along the axon. Okay, now at rest, when there's no electrical impulse being fired, okay, so there's no stimulus, then the outside of the axon is positive relative to the inside. This does not mean that the outside is, let's say, plus 50 and the inside is minus 30. It means that the outside is more positive than the inside. So it could mean that the outside is plus 150 and the inside is plus 80. So it's more positive than the inside of the axon. Okay, and that's brought about by the sodium and potassium channels being closed. Now some they do leak slightly, but mainly they're closed. But the sodium-potassium pump will kick in the resting membrane potential. It transports 3 sodiums, 3 positive ions out for every 2 potassiums or two positives in. So 3 positives going out, 2 positives going in. So therefore the outside is positive relative to the inside of the membrane. Okay, again, that's shown by this diagram here. So we have our sodium-potassium pump breaking down ATP, using that energy to actively transport your 3 sodium ions out for every 2 potassiums in. So outside more positive than the inside. Okay, and again, here's your resting membrane potential. So that's the cross section through your axon. Again, positive on the outside relative to the inside of the axon. Okay, now when there is a stimulus, then we talk about an action potential. Okay, so the action potential, that will be caused, as I say, by a stimulus, which will be detected by receptors. Those receptors cause sodium ions to open. Okay, and because you've got lots of sodiums on the outside compared to the inside, then sodium ions flow into the center of the axon. Okay, and that depolarizes the axon. Okay, so it means that the axon at that point where the sodium channel is open is positive on the inside compared to the outside or relative to the outside. Okay, so more positive on the inside. So that's your depolarization. And then these further sodium channels along the axon, or what we call gated channels or voltage gated channels, they realize that there's depolarization occurs here, which causes them to open, depolarization, next one opens, depolarization, next one depolarization, etc., etc. So you get that sort of domino effect or Mexican wave effect of depolarization passing down along the axon. Okay, so positive on the inside relative to the outside. Okay, and you can see that here, your sodium channels open, sodium ions flow in through those sodium channels, causing the inside to be more positive to the outside. Okay, that will reach a threshold, which causes then more sodium channels to open, so you get total depolarization of the axon. Okay, then you get repolarization. Okay, so that's it becoming more positive on the outside again compared to the inside of the axon. And that's brought about when potassium channels open. Okay, so around about plus 40 millivolts, those sodium channels will close. That causes potassium channels to open. Potassium flows out again in that sort of domino effect, those voltage gated channels along the axon, opening slightly after one another. And that will cause depolarization, so potassium channels moving out through the axon to the outside, more positive on the outside again, relative to the inside. Okay, and again, here you can see that so your sodium channels close, potassium channels open at about plus 40, and potassium channels, or potassium ions, sorry, flow out to the outside of the axon. Okay, so we get this action potential sort of graph. Okay, so normally your resting membrane potential is about minus 70. Here's some of the a receptor picking up a stimulus, some sodium channels opening. If that then reaches a threshold value around about minus 40, that causes, or minus 40, a lot of sodium channels to open, which then causes that depolarization. Then at plus 40, the sodium channels will close, potassium channels open, potassium ions flowing out to make it more positive on the outside again, so that repolarization. There's a slight overshoot or hyperpolarization here, where it takes the potassium channels a little bit longer to close. Then the sodium-potassium pump kicks in again and brings about the resting membrane potential. And that is described in this little animation.
When the cell membrane is at its resting membrane potential, the activation gates of the voltage-gated sodium ion channels are closed and the inactivation gates are open. Voltage-gated potassium ion channels are closed. Depolarization is initiated by a stimulus which makes the membrane potential more positive, causing the voltage-gated sodium ion channels to start to open. As threshold is reached, many sodium channels open. Sodium ions diffuse across the membrane, causing depolarization. Voltage-gated potassium ion channels also begin to open, but more slowly. Therefore, depolarization occurs because more sodium ions diffuse into the cell than potassium ions diffuse out of it. As the membrane potential approaches maximum depolarization, the inactivation gates of the voltage-gated sodium ion channels begin to close, and the diffusion of sodium ions decreases. The potassium ion channels remain open, and potassium ions continue to diffuse out of the cell. The increased potassium ion permeability lasts slightly longer than the time required to bring the membrane potential back to its resting level. The extra efflux of potassium ions causes the membrane potential to become slightly more negative than the resting value. After the voltage-gated potassium ion channels close, the active transport of sodium and potassium ions reestablishes the resting membrane potential.
Okay, we can also see that in this little animation or this little PowerPoint. So at rest, you've got your sodium-potassium pump taking out those sodium ions, 3 sodiums for every 2 potassiums coming in. So more positive on the outside compared to the outside, compared to the inside. Okay, when stimulated, so some form of stimuli is picked up by receptors, the sodium channels open and sodium ions flow in, causing that depolarization of the axon, so more positive on the inside compared to the negative on the outside. Okay, that will then pass on. As you can see, these sodium voltage-gated channels further down your axon then open, and that causes your sodiums to move in, so you gain depolarization further down the axon. After that, after the depolarization in this section, you get repolarization, so your potassium ions moving out, and then you can see the sodium-potassium pump kicking in again to bring it back to your resting membrane potential. Further down the axon again, you've got depolarization here, repolarization in this section, and that continues in that sort of domino effect as we talked about. So sodium ions moving in, potassium ions moving out, sodium-potassium pump bringing about that resting membrane potential again, and that's sort of seen as the refractory period. Okay, so as we can see here, that domino effect of sodium and potassium ions moving in and moving out, and then bringing about resting membrane potential again. Okay, moving on to the next bit, we talk about a threshold stimulus. Okay, so here you can see different intensities of a stimulus. Okay, so these first 3 don't produce an impulse. Okay, that perhaps is your threshold along here. These next 3, which get above threshold, do produce a stimulus. So that's something known as the all or nothing law. Okay, if it doesn't reach threshold, no impulse is sent. If it does, then an impulse is sent. It's a bit like if you're sitting outside and you can see a fly landing on your hand, you can't actually feel it because the stimulus or the mass of the fly doesn't reach that threshold value, so you don't get that impulse being sent. Okay, in terms of propagation of the impulse, as we talked about, that domino effect, your impulse is moving to the left here, you can see that depolarization, then the repolarization just after it, and that will cause depolarization here, the next bit, next bit, next bit. So your impulse passes down the axon. Okay. Now, also in terms of the propagation of the axon or the impulse, the myelin sheath helps with that as well. Okay, so the myelin sheath blocks these channels, if you like. It's an insulating layer which forms around, or the cells provide that insulating layer around the axon. If you think about these channels that you have in here, they're out of action. Okay, so the only sodium-potassium channels that will be able to open and close are at the nodes of Ranvier. Okay, so in this case, you get depolarization in the node of Ranvier, then that will jump to the next node of Ranvier, next node of Ranvier. So the impulse sort of jumps between the nodes, which causes that impulse to speed up or be faster along the axon. Okay, another thing that can affect that is the diameter of the axon, and that's simply to do with the number of channels. Okay, smaller diameter, less channels around that diameter, less influx of ions or outflux of ions. Whereas when you have a wider diameter of axon, you have more channels which can become depolarized, repolarized. So therefore more ions can move in and out, and that allows the impulse to travel faster as well. Temperature is another thing. That's simply to do with the diffusion of these ions in and out. So the temperature increases, more kinetic energy, these ions can flow in faster, which then causes the impulse also to travel faster. Okay, the last part of this is then looking at the synapse. Again, this diagram just out of the textbook. So at the synapse, that's just the gap between, let's say, the sensory neuron and the connector neuron. Okay, or connector and motor neuron is the gap between the two neurons. Okay, this is often referred to as the synaptic bulb, which leads, this is the pre-synaptic neuron, so the impulse is traveling from left to right. Synaptic bulb, synaptic cleft is the gap, and then the postsynaptic neuron, which has these receptors for the neurotransmitter. Okay, and the way this works is that if your impulse is traveling down from, let's say, a receptor down your sensory neuron, the arrival of the impulse at the synaptic bulb causes calcium ions, or calcium channels, sorry, to open. Okay, and that causes an influx of calcium ions into the synaptic bulb. That causes these synaptic vesicles, which contain some neurotransmitter, to move towards the synaptic cleft. Okay, the neurotransmitter is often ACH, or acetylcholine. It then is dropped off, if you like, by exocytosis in the synaptic cleft. So these vesicles will bind with the membrane and drop off that neurotransmitter. The neurotransmitter at this point of, or this part, is at high concentration, and it diffuses across the synaptic cleft. So high concentration to low concentration, and it will bind with receptors. Okay, if enough of that neurotransmitter binds with the receptors, then it causes what we call an excitatory postsynaptic potential, or an EPSP. If that reaches threshold, then it causes sodium channels to open along the axon, and we get our impulse traveling down the next neuron. So it has to reach threshold at EPSP for an impulse to be initiated in the postsynaptic neuron. Okay, in this case, if our neurotransmitter is ACH, it requires acetylcholine esterase, or ACHase, to break down that ACH into choline and ethanoic acid. It's transported back into the synaptic bulb, and ATP from these mitochondria is used to resynthesize that ACH. Okay, so just go through that again.
Action potentials arriving at the presynaptic terminal cause voltage-gated calcium ion channels to open. Calcium ions diffuse into the cell and cause synaptic vesicles to release acetylcholine, a neurotransmitter molecule. Acetylcholine molecules diffuse from the presynaptic terminal across the synaptic cleft and bind to their receptor sites on the ligand-gated sodium ion channels. This causes the ligand-gated sodium ion channels to open and sodium ions diffuse into the cell, making the membrane potential more positive. If the membrane potential reaches threshold level, an action potential will be produced.
Okay, so just to look at that in a slightly different diagram, you've got your impulse traveling left to right, calcium ions passing in as these channels open. The neurotransmitter vesicles drop off their neurotransmitter to bind with that membrane. High concentration to low concentration neurotransmitter passes across the synaptic cleft, binds with receptors. Sodium ions then can move in, causing depolarization in the post-synaptic membrane, and your impulse carries on. Okay. In terms of the functions of synapses, they allow that nervous communication to carry on between neurons. Okay. They also allow the movement of the impulse in one direction. Okay, so it can only move left to right. It can't move in the opposite direction. That's because you only have the neurotransmitter in the synaptic bulb, where it's resynthesized. So it can only pass from high concentration to low concentration, so presynaptic to postsynaptic neuron. And also the receptors are only found on the postsynaptic neuron, which means the impulse can only travel in that direction. Okay, it prevents overstimulation, which would mean too many impulses traveling in a short period of time. This allows a little bit of time or a little bit of a gap for the ACH to be resynthesized before another impulse can be sent. Okay, and they allow integration, which essentially means that you have many synapses with different neurons. Okay, so if you have neurons running down your leg, you may only need a few neurons running down to your left toe or your left big toe or whatever. It means we have less neurons. So it provides integration. Okay. Then they talk about summation. Okay, so summation is just where you get the additive effect, or two neurons adding together. This is sort of your integration as well. So you have a number of synaptic bulbs here synapsing with a single neuron. Okay, in terms of summation, these two could be excitatory neurons, so they deliver a certain amount of neurotransmitter. If enough of that neurotransmitter is released by both of these neurons, then your EPSP or your impulse can then travel on because it has reached threshold. Okay, some neurons are inhibitory. Okay, so here you have an excitatory neuron, here you have an inhibitory neuron. Okay, and that inhibitory neuron is there to stop the impulse being sent. Okay, so you might get neurotransmitter released from this excitatory one. You'll also possibly have neurotransmitter being released by this inhibitory one. If enough of the inhibitory neurotransmitter is released, then it prevents the EPSP and it prevents the impulse being sent in the postsynaptic neuron. So it dampens down that effect of the excitatory neuron. Okay, GABA is an example of an inhibitory neurotransmitter, and it's released to reduce anxiety and prevent panic attacks and dampen down those pathways in the brain. Okay, and then the last part of this is looking at different substances and what effect they have. So you can see nicotine stimulates the release of ACH, so that will mean that more action potentials are likely. Okay, so that makes your nervous system a bit more awake, if you like. Curare is a neuromuscular or blocks receptors at neuromuscular junctions, so that prevents synaptic transmission and loss of muscle function. So Curare, if you imagine those films where you see people that live in the forests where they blow a dart through a pipe and it knocks the person out, that is using that Curare. Often that could be using that Curare and it loses muscle function. Okay, opioids are certain drugs that will block calcium channels, and they are there to provide pain relief. So some of those can be used in operations and things where pain receptors can be blocked and reduce the amount of pain that perhaps the patient is under. Okay, hopefully that helps with neurons and synapses.